All right, so Bio30S. It's just a recording. I'm going to try this out for our notes for the first unit here, the human body and homeostasis. So we're going to look at the cells and cellular function. So we've gone over in class. So if you've already been in class, you've seen this. But anyways, we need to understand the various parts of the human body and homeostasis. In particular, you need to be aware that all things, whether you're living or non-living, are created from various chemical elements. So if you've ever seen the periodic table before, every single thing on that table makes up every single thing in the universe, with the exception of a vacuum, which is a space that doesn't have anything. When we're talking about biological organisms, you need to be having carbon, hydrogen, oxygen, and nitrogen in particular. If you organize these into a cell, you're going to be able to distinguish the difference between different cells as well, because that organization is going to be different for each and every single type of cell. The cell itself is going to be the smallest unit of matter, and that has to be able to carry on a process of life. So there are smaller things in a cell, but if we're going to talk about the process of life itself, then you have to have a cell to be the smallest piece. So there's a lot of history behind that. I'm not really worried so much when you look into the history. We've got Schleiden, Schwann, and Virchow that had this idea that things are made up of cells, and it ended up becoming cell theory. Um, there's a couple of ideas to cell theory, and I don't really need you to know about Schleiden, Schwann, and Virchow, but understand that cell theory itself, it deals with all living things being composed of one or more cells. If that's the case, you could have something that is single-celled or unicellular, or you could have things that are like us, they're multicellular organisms. If you're talking about cells themselves, they have to function together in an organism. So they have to have various clumps of cells doing certain jobs to make an organism exist or to function. And lastly, you can only get cells from reproducing cells that already exist. So you can't just magically clap your hands and create a cell. You have to have a cell that reproduces itself to turn into another one of your cells. So if you want to see a picture of it, I got one on the screen over here. Um, as you can see, there's a whole bunch of different variations and sizes of all sorts of things. At the very bottom, you can see that atoms and small molecules, or even subatomic particles that are smaller than atoms, can exist. If you get bigger, you get into actual things that we can see with our eyes. Those are obviously things that we can observe as existing. But somewhere in the middle, you got these things called cells, and they're between 1 to 100 micrometers in diameter. So you need a microscope to actually see those sorts of things, and that's things like the actual cells themselves or the specific parts of the cell as well. But before we talk about that, maybe we should understand what those parts are. Now, when we're talking about the cells, we have to understand the difference in their size. It's a good idea to note that a cell can only be a certain size. You can't get big cells, and if you could, you'd be able to see cells if that makes sense, right? Now, you can see some. The human egg, for example, the ovum is the largest human cell. This is something you can see, and if you saw it, it's kind of like a grain of sand. If there was one sitting on a table or on something that you can see essentially. You can see it with a microscope if you want, but you don't need the microscope. Beyond that, though, most of our human cells are very, very, very tiny. You're going to need some sort of assistance to see any of them. They have to be small for a reason, and there's a couple of reasons for that. The first one is that there's only a certain amount of volume that a cell can maintain and hold. Think of it like a balloon. You can blow up a balloon, and at some point, it's going to get so big that it's going to pop, and that's no good. That's not really useful to us. Same thing with a cell. You can only get it filled so much with cytoplasm before it's going to pop. So it's not useful to have a very large cell. The volume has to be a certain size. The other part is the surface area ratio. It might be hard to understand, but I've got an image at the bottom, and I apologize. It's a little bit smaller, but there's 3 cubes there. The smallest one has a surface area of 6 units squared. So in this case, they're millimeters, 6 millimeters squared, and it has a volume of 1 millimeter cubed. So you can see that there's 6 active sites if you look around the cell, the active area, and the volume itself is just 1. As the cell gets bigger, though, you start to notice that the volume also gets bigger, and eventually the volume becomes more significant than the surface area itself. So if we just double the the dimensions for one side of this cube, you end up having 2 millimeter sides, and you get a surface area of 24. which makes reasonable sense. But the volume has gone up to 8 millimeters, so it has increased significantly more. The surface area has increased by a factor of four. 6 times 4 is going to give us that. Is it 6 times 4 that gives it 24? Yeah, 6 times 4 gives it 24. Sorry, I'm doing math in biology, so my brain's not totally functioning there. Yeah, surface area goes up by a factor of four. Your volume goes up by a factor of eight. So you can see if we go one step further, you have sides that are going by 4 millimeters instead. Well, you got 16 millimeters for one side, and 16 times 6 is going to give you what, 96 millimeters squared. Your volume, on the other hand, is going to be 4 times 4 times 4, which is basically 16 times 4 ish, right? And that gives you about 64. So you can see the volume is quickly getting larger and larger and larger, far more significantly than the surface area, which is only going up in small increments. Eventually, you get a situation where the volume is much larger than the surface area, and that's not useful to us. So, if that's the case, we have too big of a volume. It's harder to interact as much with those large cells. Think of it like this instead. The example I use in class is if you're doing a job, you might go to work and you might be the best at your job. So let's say something simple. Let's say you're working in a fast food chain. You got to work the cash, you got to work the fryer, you got to work the grill, you got to count the cash at the back, and you have to clean the store. You might be the best at your job, and you can do all of those jobs yourself. If you're like some superhuman, you would do it. The unfortunate thing is at the end of the day, you're going to be exhausted. So you might have run the best store ever, but you're exhausted at the end of the day. You've done a really big job. Now, like a cell, instead, what if you had a lot of small components instead? What if you got someone that is really good at working the fryer, really good at working the grill, someone who's really good at doing cash, really personable, someone who's good at counting the cash, someone who's good at cleaning? Now, instead, sure, it takes a little bit more energy, but you're going to feel pretty good at the end of the day because you're not overworking yourself and your work is going to run more smoothly because you have specific roles in that job. It's the same thing with a cell. It makes more sense to have a whole bunch of small cells doing various parts instead. So as the salt cell gets larger in volume, as we talked about, the volume actually starts to decrease. Sorry, as the volume gets larger, the surface area starts to decrease. Same idea. If you're working a job, the more jobs you do, your productivity is going to start to decrease as well. So it makes more sense to have smaller pieces. It also limits how much you can move, right? So the bigger you become, the harder it is for you to move around. It makes it harder to metabolize the cells as well. So it makes it difficult for the cells to actually have things to metabolize. If you have a greater surface area, you might need to have a modification. So things like microvilli, the little feelers on the cell, or you might fold a little bit like red blood cells do, or you might develop a flagellum to propel yourself from place to place like a sperm cell does. But you might need these additional pieces if you're functioning as a cell. The cells themselves, they have different shapes. So I got a couple of pictures on the side there. There's tons of other ones, but depending on what you're working with, you're going to have a different type of cell. So nerve cells, they're sending an electrical signal. It makes sense that the fibers of a nerve cell, a neuron, look like a lightning bolt. It's really easy to send electrical signals that way, and they're very long because you're sending electrical signals all throughout your body. A red blood cell, on the other hand, you can see it's in the top left of that picture, is very smooth and round, and it makes sense as well because a red blood cell has to move through your blood vessels without getting stuck or caught on anything. So having them small and smooth and round makes it easier for them to go through the various blood vessels that you have. Depending on what kind of cell you have, that's always going to change as well. You got muscle cells, diatoms, parameciums as well. Um, Depends on what you're working with. Just a big look at the red blood cell there. We look at the actual inside of the cell to understand what is working and how the cell is working itself. Just like we have organs, our cells have what are called organelles. An organelle is a component that does a very specific job for the cell. So just like we have organs in our body that do a very specific job, our cells also have organelles that do very specific jobs as well. And it's the very same idea as us. They need to maintain the life of the cell. There's a couple of features that most cells have in common. Not all cells, but most cells will have this in common. One of those things is a cell membrane that keeps the cell enclosed so that the goop doesn't come out of the cell. Um, Inside the cell, there's a whole bunch of organelles. Those organelles should have their own membrane as well. You don't want those merging with each other. So We don't have like a soup of our heart and our liver and our pancreas all in one go. Instead, we have specific spots. We have a heart, we have a liver, we have a pancreas, so on and so forth. Your cells have that as well. Right near the middle of the cell, usually around the middle, you're going to have what's called a nucleus. That's kind of the brain of the cell. It doesn't have to be right in the middle, but usually it is. That's usually going to be in most cells. Not all cells, but most cells will have that nucleus as well. If you don't have the nucleus, you're a different type of cell. If you do have the nucleus, you're what's called a eukaryote. If you don't have it, we'll call you a prokaryote. We're not going to talk too much about the prokaryotes in the grade 11 level course. If you take the grade 12 course, you'll learn a little bit more about some prokaryotes. Those are things like bacteria and bacteriums. They basically have a function, they have to do their job, and that's how they exist. Whereas if you're talking about eukaryotes, plants, mammals, fish, all that fun stuff, those are eukaryotes. So we're a lot more similar to trees in that regard than we are to a bacteria. A quick look at a cell here. I'm not going to go over everything just yet. I have some dedicated slides for that. But if we were to look at the cell, you can see it's kind of overwhelming. It's like a city. You've got a nucleus that's kind of like this purple gumball on the side. The cell itself has a whole bunch of these hairs on the outside. Those are cilia. And then it's got a whole bunch of other pieces that have to function to make the cell work. On the topic of the various pieces, we're going to jump straight into a couple of them in particular. So we talked about the eukaryotes, the animal cell. In the animal cell, you're not going to get any of the same. So my human cells are not going to be same as, say, a pig cell or a bat cell or something like that. But they're going to have some similarities. And there's three things. Typically, they'll have a nucleus. You should have cytoplasmic organelles, which means your organelles, the organs inside of the cell, are floating around in the cytoplasm. You should also have a cell membrane that encloses the cell also. So it's holding everything together. That usually should exist, not always, but usually it should. The nucleus is the brain of the cell, so that's the first thing we're going to try to understand as well. The nucleus is the most prominent structure. If you look at a cell in a microscope, you'll see a big black dot. That's usually the nucleus that we're looking at. It maintains its shape with a protein skeleton. We call that the nuclear matrix. And the nucleus is essentially the brain of the cell. It's going to make sure the cell does what its job is. It has all the genetic information and the makeup in the cell also. Inside of the nuclear envelope or on the nuclear envelope itself, you got these teeny tiny holes, and that allows proteins to come in and out and various chemical messengers so that the nucleus can send its signal to wherever it needs to go. You also have the DNA, which is inside of the nucleus. Big blown up picture of what that looks like. You can see the purple part there. That is our nuclear envelope. Nuclear pores. You got chromosomes inside as well. And right in the middle of the nucleus, it's like a brain within the brain called the nucleolus. We'll talk about that right away as well. So the chromatin is that thread-like material that coils into the chromosomes before cell division is going to happen. The chromosomes are the rod-like structures, and that's formed during cell division. That is your genetic material, or that's where your DNA is. The nucleoplasm is a semi-fluid medium of the nucleus. So it's kind of like the cytoplasm in the cell, but it's a fluid specifically for the nucleus. Inside, there's also the nucleoli. That is these little tiny dark spherical bodies, and it's where the ribosomes are produced. So if you ever want ribosomes to create proteins, you produce them in the nucleoli. They end up going through those nuclear pores, and those nuclear pores will allow those nucleoli to get to the endoplasmic reticulum, which we'll talk about in just a minute here. The envelope that surrounds the nucleus, this is a double membrane, so it's like having two security guards outside of the nucleus. You don't want just anything coming into your brain. So as humans, we have what's called a blood-brain barrier to prevent things from getting to our brain. In our nuclear or in our cells, our nucleus has a nuclear envelope. And then you got those pores, which are about 100 nanometers. So nothing really can get through 100 nanometers. It's very small, with the exception of a couple of very specific pieces, proteins in particular. That's going to be a bit of a theme here. Proteins can get in, ribosomes can get out. The nucleus also has that DNA that's going to determine the characteristics of each individual cell. Eventually, we get to the endoplasmic reticulum, which kind of surrounds the nucleus itself, and it's a really good spot because the rough ER, which is the closest to the nucleus, is going to attach or allow the ribosomes to attach here. That's why it's called the rough ER. If you were to see it, it looks very speckled. It's got a whole bunch of ribosomes attached to it. So all over on the side, you take a picture of it or take a look at it, you can see the nucleus is that purple section in the back. The pinkish reddish area is our endoplasmic reticulum. The rough part is the one with all the studs on it, the pinkish reddish part. The smooth part, the SCR, is right after the red part, that peachish orangish color. There's no ribosomes in the smooth endoplasmic reticulum. So the smooth is continuous. It just connects to the ER or the RER, but it doesn't have the ribosomes. So there's no protein synthesis happening here. Instead, you get detoxification and a little bit of short-term storage. It creates what are called transport vessels, and the vessels are going to go to somewhere else. The smooth ER has a whole bunch of enzymes as well, which are there for fat metabolism. So they help you break down fats. And it's there for the synthesis of non-protein steroid hormones. So it can produce some steroid hormones for us, similar to things like cholesterols. The amount of ER is kind of interesting because that depends on how much a cell is working. So this is why people that are fairly active and always on the go, 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 they tend to be slimmer in a sense. This isn't like perfect logic, but they tend to be slimmer because their body is able to metabolize more fats because their body produces more or has more endoplasmic reticulum in their cells. So you can metabolize more fat. The catch here is if you're functioning more, if you're doing more work, you're moving around more, you're probably eating more and consuming more fat anyways. So there is a balance. It doesn't mean that because you go for a jog every morning, you can eat donuts for breakfast. What it means is that you can consume more, but you can also metabolize more. The opposite is also true. People that don't engage in this much activity, people that tend to sit on the couch all the time, they're more likely to get that fat storage building up on their bodies because they can't metabolize as much of that fat. So they end up eating less, or they should be eating less because their cells just can't deal with that. And instead, the fat goes somewhere else. It gets deposited as adipose tissue, and it's just stored typically in your gut. Go back to the ribosomes, though. This is where proteins are going to be made. They are protein factories. They pump out a lot of them. They have two parts to them. They have a large and a small subunit, and they work through DNA specifically. So mostly a ribosome is just RNA and some DNA, and they just transcribe the information and create what is called a protein. Typically, they're attached to the endoplasmic reticulum, but they can float around in the cytoplasm as well. We see a quick picture of a ribosome zoomed in. That's kind of what it looks like. You have the mRNA that's coming in through the bottom. It connects to the tRNA where it's transcribing the information, and that is telling the protein or telling the ribosome to create a protein, which is just a chain of amino acids. So a protein is just 3 or more amino acids hooked up to each other, and the ribosome will be told specifically which amino acid to create which protein to create, and it will find those amino acids and connect them together. Because proteins are so simple like that, that's why we can make artificial proteins. That's why you can go to a health food aisle and buy protein powder, is because if you know the amino acids, we can just connect them ourselves and create protein that way. This is called protein synthesis. The word synthesis means to create. Protein synthesis means creating proteins, essentially. The ribosomes on the ER, they depend on a very specific sequence to bind to a receptor protein, and that's what's going to create the various proteins themselves. That brings us to the Golgi apparatus that comes right after the ER, named after this cool dude named Camillo Golgi. It's very close to the nucleus, and it's just a whole bunch of these little sacs, little pockets that can store various content. So it's kind of like the UPS of the cell. If you need to get something from one place to another, so let's say you created a protein and you want to deliver that protein to another cell, you need to get it to UPS to deliver it for you. That's the Golgi apparatus. So your cells will send the information or it'll send the proteins to the Golgi. And what it's going to do is it's going to package everything into a sac for you, and then it will be delivered to another cell from there. All of the proteins from the ER will end up here eventually. They're going to fuse with the Golgi apparatus. They'll process, they'll package, and then they will secrete that into the cell and then wherever it needs to go from there. That leads to the lysosome. These are security guards in our city cell, or however you want to look at this metaphor. They're made by the Golgi apparatus, so believe it or not, in this case, your security guards are coming from UPS. They are just pockets full of digestive enzymes. They pinch off, and then they look for whatever shouldn't be there, whatever they can mess up a little bit, and they will look for, say, a food vacuole, and they will fuse with it and then break it down with the various enzymes that it has. So if you want to see a picture of what that looks like, right over here, we've got a chunk of a cell, and you can see a food vacuole. This is this brown bit on the left that is entering the cell, and you can see it's just a pocket with a food vacuole or food pocket in there. The lysosomes are pinching off of this pink Golgi apparatus. The lysosomes are the white bits with the red bits inside. The red bits are the digestive enzyme. So they fuse with the food vacuole. They will break down that food vacuole. Eventually, it has been digested, and then the cell will eject that. That is called exocytosis, and then eventually elimination as well. The lysosomes can also undergo something called autodigestion, where it eats part of its own cell, or it can help with apoptosis, which is programmed cell death. Your cells have to kill themselves after they've done their job. So what they can do is they can they can reabsorb a tadpole tail so that your cells can't move anymore. If they can't move, they can't function. They'll die. Um, if there's ever an issue, it can create something called degeneration of webbing between human fingers. So, usually when you're a fetus, you'll grow with little webs, kind of like a frog. But through apoptosis, you should develop a situation where that webbing gets destroyed. Some people still have web fingers and toes because the apoptosis didn't function properly there. If you don't have proper lysosomal enzymes in your body, you'll end up having some childhood diseases to compete with. Our medicine is pretty good, though, here in North America, that typically you'll survive in some way. But there are some instances where that won't actually work out for the better, which kind of sucks. But we're talking about biology here, so we can understand that that can happen. We also got these things called peroxiomes. These are membrane-bounded vessels that contain very specific enzymes. These are usually found in the liver, which is your detox site. They create hydrogen peroxide, so that comes in that brown bottle. If you ever get a cut, you might put it on your skin, and it stings a lot. That breaks down into water and oxygen by an enzyme called catalase. In old dying cells, the membranes of lysosomes are broken down. They free the digestive enzymes into the cytoplasm, and it just kills the entire cell, which sounds pretty brutal, but that's a good thing. You want the cell to die when it gets old and replaced with something that is newer. That leads to a vacuole. A vacuole is a large sac. It's just an empty space. Depends on where you're dealing with a vacuole. So if it's a plant cell, the majority of the plant cell is a vacuole. If it's an animal cell, you have little small vacuoles in there. So as I mentioned, they're pretty prominent in plant cells. About 60% of the cell. If you're talking about a cactus or an orchid, it can be as high as 80%, which makes sense. They just store water in a plant cell. So plants don't have feet. If they get thirsty, they can't just walk to the sink and get some water. They need to store it in their cells. So they store it in their vacuoles. Humans and animals, on the other hand, we might have a whole bunch of different types of vacuoles, maybe digestive ones or water regulating ones. We can typically get a glass of water if we're thirsty, but we still store a teeny bit of it in our vacuoles just in case we can't get to water right away. So if you want to see a picture of it, there you are. There's your cell, big pocket. That is your vacuole. And that's going to lead us to the mitochondria. That's as far as we got with the morning class at the time of this recording. So I'm actually going to pause here. I'll do another recording continuing on this a little bit later on. But this should get you up to date for what we covered on February 10th and 11th. And then we'll pick up on things from here.